Just to orientate everybody, this would be inferior, this would be superior towards the upper part of the brainstem. Here we would have anterior and posterior towards the cerebellum. So starting inferiorly, we can observe the medulla oblongata over here, and we can see the two pyramidal tracts on either side with the olivary nuclei projecting out towards the lateral surface of the medulla oblongata. Here we can see the pons, and we can see the groove of the pons in which the basilar artery would lie, and we can see the pons reaching laterally, projecting in towards the cerebellum via the middle cerebellar peduncle. As we move superiorly, we can observe over here the basis pedunculi, within which we would find the substantia nigra. And if we move more superiorly, we can see the two mammillary bodies over here, marking the posterior boundary of the hypothalamus. And we can see anteriorly there's this area that is interrupted. We would find here in the brain in situ the pituitary infundibulum reaching into the sella turcica, which it would then connect to the pituitary gland. So this would mark the anterior boundary of the hypothalamus. Moving anteriorly, we can see the two optic nerves reaching in towards the optic chiasm, which we can see over here, and then leading towards optic tracts, which would then lead towards the lateral geniculate nuclei, and then project via the optic radiations in the temporal lobe to the occipital lobe, to the primary visual cortex. Here we can observe the olfactory nerve. We can see the olfactory tract over here and the olfactory bulb, and we can see the olfactory trigone, which is formed by the medial and lateral olfactory stria. Here we can see the orbitofrontal cortices. Here we can see the medial aspect of the temporal lobe, and very importantly the parahippocampoid gyri on either side, which appear to hug the brainstem. Here we can see the uncus, and deep within this fold of the parahippocampoid gyrus we would find the amygdaloid nucleus. Here we can observe the lateral surface of the brain, and just some basic landmarks. Over here we can see the Sylvian fissure, and the Sylvian fissure separates the temporal lobe from the frontal lobe. We can see here the central sulcus, which runs from the dorsal aspect of the brain down the lateral surface, and it runs towards the Sylvian fissure. And we can see anterior to that a very distinct gyrus, the precentral gyrus, and over here we can see the postcentral gyrus. So the precentral gyrus would be the primary motor cortex, and the postcentral gyrus would be the primary sensory cortex. Just anterior to the precentral gyrus, we find a thin, indistinct area that's not really bounded anatomically, but functionally this is known as the motor association area. And the same on the posterior aspect of the postcentral gyrus, which would be the primary sensory association area. The frontal lobe is designated by, or at least bounded posteriorly by the central sulcus, and it then reaches anteriorly and inferiorly bounded by the Sylvian fissure. Over here we can see the operculum, which is formed by the inferior frontal gyrus. And the operculum has several distinct areas. We have the orbital part over here, the pars triangularis and the pars opercularis. And this area would then form the well-known Broca's area. If we proceed to the posterior aspect of the Sylvian fissure, this area over here would be Wernicke's area. Wernicke and Broca's area are connected by a deep white matter fasciculus running underneath the cortex over here, known as the arcuate fasciculus. So Broca's motor speech area communicates with Wernicke's auditory association area. The primary auditory cortex we find in the superior temporal gyrus in the posterior aspect of the Sylvian fissure. If we look at the posterior aspect of the brain, just to view the primary visual cortex, here we can see the occipital lobe And the primary visual cortex is essentially formed on the medial surface by the calcarine sulcus. We have the primary visual cortex, which is area 17 of Brodmann, and then we have the visual association areas, which cover a stepwise increase in surface area from posterior medial towards the lateral surface of the brain, which would be area 18 and area 19. I'm going to cut the brain at midline so that we can see the midline structures. So I'm inserting the brain knife into the interhemispheric sulcus, and I am aligning the knife with the optic chiasm anteriorly, and I hope to proceed through the two mammillary bodies. So here we see the hemisected brain, and we have a sagittal midline section. Just to orientate everybody, that's anterior, posterior, inferior, superior, and this is the medial aspect. Starting inferiorly, once again, with the brainstem, we see the medial aspect of the medulla oblongata. And moving superiorly, here we see the expansile pons, and the pontomedullary junction over here. As we proceed further superiorly, we can see the midbrain over here, and projecting laterally, we see the basis pedunculi over there. Posteriorly, we observe the medial surface of the cerebellum, and we can see the white matter as it reaches into the cerebellar hemisphere, which we refer to as the arbor vitae in Latin, which is the tree of life. Here we can see the individual gyri of the cerebellum, and these are the folia, which in Latin is the leaves of the tree of life. As we look at the midbrain, we can see that the midbrain is divided by this conduit, which is the cerebral aqueduct of Sylvius. Now, the cerebral aqueduct of Sylvius connects the fourth ventricle, which we can see over here, with the third ventricle, which we can see over here. The cerebral aqueduct of Sylvius divides the midbrain anteriorly into the tegmentum and posteriorly into the tectum, and the tectum is composed of the superior and inferior colliculi. So if I just remove this pia, we should be able to see the superior colliculus and the inferior colliculus over there. The normal midbrain has a convex border, which we can see over here, and we can see the wall of the third ventricle, which is essentially formed by the thalamus, the medial aspect of the thalamus. Anteriorly, we can see the mammillary body. So the mammillary body marks the posterior boundary of the hypothalamus, and we can see this groove over here, which is the hypothalamic sulcus, and this designates hypothalamus from thalamus. Within the walls of the hypothalamus, we find the hypothalamic nuclei, which are the functional unit of the hypothalamus. Here we can see the optic chiasm, and we can see the anterior commissure over here and the posterior commissure over here. Attaching onto the posterior commissure, we can see the pineal gland, and stretching from one thalamus into the other is the intrathalamic adhesion, or the massa intermedia, as it's sometimes referred to. So what I'd like to do now is demonstrate the midbrain structures that are implicated in affect. And just to orientate everyone again, this is the brainstem. So we can see the medulla oblongata over here, the pons with the middle cerebellar peduncle leading into the cerebellar hemisphere. Here we can see the midbrain. So I would like to make a section through the superior colliculus so that we can observe the substantia nigra. So using the brain knife, I'm going to cut in that plane. and then while still observing the orientation of the cerebral aqueduct, the superior colliculus, which has been transected through, inferior colliculus, cerebral aqueduct, tegmentum, we then rotate the brain. So just to orientate everybody, now we can see an axial section through the midbrain. This would be anterior, posterior over here. We have superior and inferior, medial and lateral, and we can observe the cerebral aqueduct, which divides the tectum from the tegmentum of the midbrain. Here we can see the basis pedunculi, which proceeds laterally away from the midline, and we can see this dark area over here. This dark area is the substantia nigra. The substantia nigra, black substance in Latin. It is pigmented by the metabolism of dopamine, which forms neuromelanin, and the substantia nigra has two component parts. It has the pars reticulata, which is on the outside, and the pars compacta, which is on the inside. Upon gross inspection, this is not possible to be distinguished. However, on histological sections, we are able to see the different aspects, and they also have different projections. Over here we can see, we'll barely see the red nucleus. The red nucleus is involved in the rubrospinal tract. It is highly vascular in the living individual. However, when perfusion ceases due to death, it is blanched out. So the red nucleus over there. If we take a closer look at the cerebral aqueduct over here, we should be able to distinguish an area of darker contrast around it. Now, once again, this is not easily seen without stained specimens. However, the periaqueductal gray matter is found around the cerebral aqueduct of Sylvius, so in this vicinity over here. If I just rotate the brainstem a little bit, you should be able to see the cerebral aqueduct of Sylvius over here in relation to the substantia nigra over there. And once again, the periaqueductal gray matter is found in a column around the cerebral aqueduct, which we can see very clearly over here. As we proceed superiorly from the brainstem and the diencephalon, we can see the body of the fornix over here, and we can see the corpus callosum over here, and stretching in between these two white matter structures is a thin layer of ependymal cells, which we refer to as the septum pellucidum, separating left and right ventricles. There is no functional aspect to the septum pellucidum. It's simply a remnant of the embryological formation of the brain. The corpus callosum, we can see the rostrum over here, the genu, the body of the corpus callosum, and the splenium of the corpus callosum posteriorly. As we move further superiorly, we can see the structure over here, which appears to encircle the corpus callosum, and it then delves inferiorly and laterally from the medial surface of the brain. This is the cingulate gyrus. Here posteriorly, we are able to see the calcarine sulcus. On either side, we would find the primary visual cortex, area 17. So as we remove the septum pellucidum, we should be able to see within the lateral ventricle. So we can now see the body of the fornix and the corpus callosum as two separate entities, and we can see the lateral ventricle exposed, and within the lateral ventricle, forming the lateral wall of the anterior horn of the lateral ventricle, is the head of the caudate nucleus, and the tail of the caudate nucleus projecting posteriorly. We can then see the lateral ventricle communicating with the third ventricle via the interventricular foramen of Monroe. We can then observe the third ventricle connecting via the cerebral aqueduct of Sylvius with the fourth ventricle, so doing facilitating the flow of cerebrospinal fluid. What I'd like to do now is I'd like to remove the cingulate cortex to reveal the cingulum. Now, the cingulum is a white matter fasciculus that gathers information from the cingulate cortex and transports it down into the parahippocampoid gyrus into the entorhinal cortex, which then feeds into the hippocampus. So I'm going to reflect the cortex just to orientate you. This is anterior, posterior, superior, and inferior. We're still on the medial surface of the brain. So I'm just going to start by reflecting the cortex. And this is facilitated by just scraping it away. And now we can very clearly see a contrast between what is the gray matter or the cortex and the white matter underneath. So we can see that the white matter also follows the same direction as the cingulate gyrus. So what I'd like to do now is remove the cingulum in situ so that we can observe what it looks like. And we can see that this is the cingulum, and we can see the white matter that it is composed of. And in its absence, we then see the void that it has left. So the cingulum encircles the corpus callosum and the diencephalon, transporting information from the cingulate cortex into the parahippocampal gyrus. Over here, I have removed the cortex surrounding the corpus callosum. We can observe the corpus callosum in isolation now and see that it projects superiorly and it projects anteriorly, posteriorly, and laterally and inferiorly. The corpus callosum, therefore, truly is the great commissure connecting the left and right cerebral hemispheres. The sum total of the corpus callosum, therefore, isn't what we conventionally think of on a midline sagittal view as this structure over here, but it really is a radiating structure which connects all crevices of left and right hemispheres with one another. So what I'd like to do now is I would like to expose the lateral ventricle so that we may observe the caudate nucleus more fully. So with the corpus callosum removed, we can now see what the lateral ventricle looks like exposed. And just to demonstrate the radiating fibers of the corpus callosum, we can see these fibers traveling from the medial aspect of the corpus callosum and radiating superiorly. So this is not the entire corpus callosum, but you can see some of the radiating fibers as they pass through the body of the corpus callosum and then radiate superiorly. You can see the individual white matter bundles as they project superiorly. The same goes for these fibers that project to the contralateral hemisphere, anteriorly, posteriorly, laterally, and in all essential directions, connecting one cerebral hemisphere with the other. So here we can observe the lateral ventricle exposed, and we can see the head of the caudate nucleus anteriorly, and we can see the tail of the caudate nucleus projecting posteriorly. We can also see the thalamus more clearly, so this is the medial surface of the thalamus, and we can see the body of the fornix projecting over it. And if we tilt the brain a little bit, we can see that the thalamus actually projects laterally into the brain, with the medial wall of the thalami forming the lateral wall of the third ventricle. We can see this fluffy structure running on top of the thalamus. This is the choroid plexus, which is responsible for the production of cerebrospinal fluid. So I'm going to remove the choroid plexus so that we can see what the thalamus looks like. So we can now see how the body of the fornix runs on top of the thalamus. So this entire structure over here is the thalamus, and we can see the caudate nucleus lateral to that, and then the stria terminalis, which is a white matter bundle, runs right in between the thalamus and the body of the caudate. So this would be the stria terminalis over here. I'm going to move to the lateral surface of the brain, and with this I am going to remove the frontal lobe so that we can see the insula within. So if I retract the inferior frontal gyrus and the superior temporal gyrus of the temporal lobe, we can see within the insula, which I refer to as the fifth lobe of the brain, hidden within the depths of the sylvian fissure. So this is the insular cortex over here, and what I'd like to do now is dissect that out. So now that the frontal lobe has been removed, we are able to see the temporal lobe, and we are able to see the insula, which is found within the depths of the sylvian fissure. So over here we can see the superior temporal gyrus, and over here we can see the insular cortex. If I just retract the temporal lobe a little bit, you can see the gyral architecture of the insula, which is over there. So deep to the insula, just medial to the insular cortex, we would find the lentiform nucleus, which is composed of the putamen and the globus pallidi. So what I'd like to do next is remove the insula so that we can see the lentiform nucleus in situ. So we're just turning the brain around. So here once again we can see the caudate nucleus. So I'm just going to separate this tissue. Remove the insular cortex so we can see the head of the caudate nucleus, and laterally we can see the insula. So now the insula is removed as a whole. So if we put that back, you can see where the insula was. All right. And if we move to the medial surface, we can see where the head of the caudate nucleus was. So now we remove the insula, and we can see underneath this structure over here. And this is the lentiform nucleus. The putamen, which is on the lateral aspect outside, and medial to that would be the globus pallidus. You can see the different contrast between the white matter that overlies the lentiform nucleus, the so-called external capsule, and you can see the gray matter of the putamen underneath, which is slightly darker. So the lentiform nucleus over here, putamen on the outside. And we can see if we put back the insula where that would be in relation to it. So we're going to remove the insula, once again, lentiform nucleus, and we can see this white matter bundle projecting out of the lentiform nucleus, which is the corona radiata. So if we look at it anteriorly, we can see the head of the caudate nucleus over here with the tail projecting posteriorly. And if we put the insula back, we can see what that looks like in situ. So once again, the head of the caudate nucleus, frontal lobe over here, insula on the lateral aspect. Medially, we can observe the head of the caudate nucleus, and laterally we can observe the putamen, which is the lateral aspect of the lentiform nucleus. And we can see that it appears that these two nuclei on the anterior view are actually communicating. So if we just orientate everyone, we can see this is superior, inferior, medial and lateral. This is a coronal view, an in-face view. We can see the brainstem inferiorly, the frontal pole of the temporal lobe. So here's the head of the caudate nucleus, there's the anterior aspect of the putamen, and this area over here, this transitional area, this is the nucleus accumbens. And the nucleus accumbens essentially connects the caudate nucleus and the putamen on the lateral surface of the brain. So we have three nuclear complexes here. We have the caudate nucleus, the nucleus accumbens over here, and the putamen which is lateral to that. What I'd like to do now is I would like to make an axial section through the brain to demonstrate the conventional orientation we usually see in books and on MRIs. Anterior, posterior, superior and inferior. Here we have medial, here's lateral. And we can see the thalamus medially. We can see the splenium of the corpus callosum and the occipital horn of the lateral ventricle. We can see the head of the caudate nucleus forming the wall of the lateral ventricle. We can see the putamen over here, and we can see the internal capsule in between the thalamus and the lentiform nucleus and the caudate nucleus. So if we put the insula back, we can then see how the insula, which is the insular cortex over here, fits onto the lentiform nucleus. And we can see what that looks like in situ. So there's the insula, and we can then see the diencephalic structures over here. So if we put back the superior part of the section, we can then see how they come together again. So head of the caudate nucleus over here, nucleus accumbens, putamen, and if we open that up, we can then see the head of the caudate nucleus, the thalamus, and the putamen over there with the internal capsule in between. The insular cortex, which would be lateral to the putamen, has been removed, and we can observe the superior temporal gyrus over here, which has been cut through. Alright, so what I'd like to do now is demonstrate the parahippocampal gyrus and the hippocampus in situ. But before we do that, I'd just like to orientate everyone. So here we see the nucleus accumbens, we can see the head of the caudate and the lentiform nucleus over here. We can see the optic nerve over there and the optic chiasm, the optic tract. Here we can see the pons, we can see the basis pedunculi, the medulla oblongata, and the middle cerebellar peduncle. So I'm going to turn the brain upside down. So just to orientate everybody now, the brain is upside down. So this is inferior, this is superior, this is lateral, and this is medial. So here we can see the medulla oblongata, we can see the pons, we can see the cerebellar hemisphere, the middle cerebellar peduncle, and we can see this structure over here. This is the parahippocampal gyrus. So I'm just going to move the brainstem aside, and I would like you to have a look at how the parahippocampal gyrus encircles the brainstem. So here we have parahippocampal gyrus, and we can see that the parahippocampal gyrus folds anteriorly, and this area over here, this little fold that we see over here, that is known as the uncus. So this is parahippocampal gyrus, and that is the uncus, which is the fold of the parahippocampal gyrus. And deep to that we would find the amygdaloid nucleus. So just to orientate everyone again, anterior, posterior, superior, inferior, medial and lateral. And here we can see the uncus and the parahippocampal gyrus, and that encircles the midbrain. So the parahippocampal gyrus is functionally known as the entorhinal cortex, and it is the entorhinal cortex that feeds into the hippocampus. The hippocampus proper, the hippocampal formation, sits in exactly the same orientation as the parahippocampal gyrus, just deep to it. So deep to this cortex we would find the hippocampal formation in exactly the same orientation. Alright, so what I'd like to do now is reveal the hippocampus in its orientation towards the parahippocampal gyrus. So just to orientate you, this is anterior, cerebellum is posterior, superior and inferior. This is the lateral surface, this is the medial surface. So I'm just going to remove the brainstem structures, and I'm going to rotate the temporal lobe. So here we can still observe the parahippocampal gyrus, and we can see the uncus more clearly now. So this bend over here is the uncus, and deep to this we would find the amygdaloid nucleus. If we rotate the temporal lobe a little bit more, just to orientate you, this is anterior, posterior, superior, inferior. This is the medial aspect of the brain. So we would find our brainstem over here. So over here we can observe the dentate gyrus of the hippocampus. Remember, the hippocampus is a series of structures of which the entorhinal cortex forms a portion. Then we have the subiculum, and then the dentate gyrus and the hippocampus proper. So here we can see the dentate gyrus, which essentially forms the core of the hippocampus. And we can see on top of the dentate gyrus this thin frill-like structure over here. This is known as the fimbria, which is formed by the alveus. Now, the alveus is a thin layer of white matter that covers the hippocampal formation. It collects all of the information sent via the entorhinal cortex into the hippocampal formation. It then projects that information posteriorly via this structure over here. So the fimbria is a condensation of the alveus, and the alveus then essentially curls up into this thick bundle of white matter, which would then eventually be the crura of the fornix. And the crura of the fornix would then continue into the body of the fornix, which would then eventually lead to the mammillary bodies. But staying with the hippocampus, if we just reflect the medial aspect over here and open it up, we can then appreciate the hippocampus. So this structure over here is the hippocampus, the hippocampal formation, and we can see it is bent anteriorly and medially, and it encircles the midbrain. We can see over here the little pes of the hippocampus, which are these little bumps on the anterior aspect of the hippocampus, and we can see that the uncus is still intact. So deep within the uncus we would find the amygdaloid nucleus. Now, the amygdaloid nucleus is very difficult to see on gross morphological inspection because it has a lot of white matter running into it. It therefore needs to be stained to be able to observe it properly. But if we look at the orientation of the parahippocampal gyrus over here and of the hippocampus itself, we see that the hippocampus follows the exact same orientation as the parahippocampal gyrus. So the parahippocampal gyrus, which is entorhinal cortex, feeds into the subiculum of the hippocampus, which then feeds into the hippocampus proper. That information is collected by this thin layer of white matter on top of the hippocampus, which is the alveus. The alveus then pushes that information medially into the structure, which is the fimbria. The fimbria then condense to form the crura of the fornix. To demonstrate the circuits of Papez, I have dissected out the mammillothalamic tract. So over here we can observe the medial surface of the brain. So just to orientate you, anterior, posterior, inferior, superior. This is the medial surface. This is the lateral surface. So we can just about see the superior aspect of the midbrain. We can see the normal midbrain convexity, the cerebral aqueduct over there. We can see the superior colliculus, the posterior commissure. Here we can see the pineal gland, and we can see the thalamus. Here we can see the body of the fornix, and we can see the corpus callosum over here with the singular gyrus. Over here it's interrupted because it was dissected out. So I want to move on to the lateral surface to start over there. So we can see over here the temporal lobe has essentially been cut away, and the frontal lobe as well. We can see the inferior temporal gyrus over here, and we can see inferior frontal gyrus over here. We can see a little bit of the insula remaining. So just to orientate you, this is anterior, this is posterior, superior, and inferior. We're looking at the lateral surface of the brain. You can see in the depths of the temporal lobe the hippocampus pointing out. So we can see the hippocampus over there. We can see the pes of the hippocampus, and we can see its orientation in terms of the diencephalon. So if we look at the inferior surface, here we can see the midbrain, the substantia nigra, the cerebral aqueduct, the superior colliculus, the mammillary body, optic tract, chiasm, optic nerve, and we can see over here the parahippocampal gyrus with the uncus over here. So if we move on to the lateral surface, we can appreciate once again how the hippocampus follows the same orientation as the parahippocampal gyrus. So if we move to the posterior aspect of the hippocampus, we can see that the alveus collects into the fimbria, and the fimbria then collects into the crura of the fornix posteriorly. And we can see the crura of the fornix as they proceed anteriorly and medially over the two thalami on either side. So over here we have the left crura of the fornix, and we can see it forming the body of the fornix. So as we rotate this around, you see the body of the fornix, and the wall of the hypothalamus has been dissected out to demonstrate the columns of the fornix proceeding from the body of the fornix into the wall of the hypothalamus, synapsing into the mammillary body, and then a tract leading from the mammillary body to the anterior nucleus of the thalamus, the mammillothalamic tract. From there, the information processed by the hippocampus and transported by the fornix is then sent via the anterior nucleus of the thalamus into the cingulate gyrus. So in this specimen, I dissected out the cingulum, but just to start off where we ended in the previous clip, we can see the body of the fornix over here delving into the wall of the hypothalamus. So we have anterior, posterior, superior, and inferior. We're looking at the medial surface of the brain. We can see the cerebral aqueduct, pineal gland, superior and inferior colliculi forming the tectum, tegmentum over here. We can see the mammillary body, the thalamus over here, corpus callosum, head of the caudate nucleus. So we can see the fornix leading into the hypothalamus, forming the columns of the fornix, leading into the mammillary body. And from the mammillary body, there is a tract that leads into the anterior nucleus of the thalamus, the mammillothalamic tract. The anterior nucleus of the thalamus then projects into the cingulate gyrus, and the cingulate gyrus then via the cingulum sends that information to the hippocampus. So here I have removed the cingulate cortex, and we can see this white matter fasciculus, which is the cingulum, which collects the information from the cingulate cortex and then sends it posteriorly, and we can see the cingulum continuing inferiorly all the way around the diencephalon. We can see these white matter fibers leading into the parahippocampal gyrus, and then from the parahippocampal gyrus it then enters the subiculum, the dentate gyrus, and the hippocampus proper. That information is then once again propagated into the alveus, which then collects in the fimbria. The fimbria connects into the crura of the fornix, the crura of the fornix, then body of the fornix. That information is sent into the columns of the fornix, synapsing in the mammillary body, once again via the mammillothalamic tract into the anterior nucleus of the thalamus, therefore completing the circuit